Hi, everyone. I'm Natalie Davidson. I'm a postdoc currently in the lab of Casey Green at University of Colorado School of Medicine, Center for Health AI. And I'm happy to talk to you all about my research project called BuDDI, Bulk Deconvolution with Domain Invariance. So I'm going to kind of zoom in on each part of the poster. So what is bulk deconvolution and why is it useful? So imagine you're given a smoothie and I tell you all the fruits inside of it, give you one taste of this smoothie. Could you guess the proportion of each ingredient? So could you tell me the recipe for it? This is kind of analogous to the problem of bulk deconvolution. So we have bulk RNA-seq samples. So I take the tissue and I grind it all up. And now I want to know what were the proportion of each cell type for all of the different samples. So what we see here is for each one of these samples, they have different proportions of the different cell types, analogous to our recipe made up of different fruits. And we care about this problem because relative proportions of cell types can be a useful biomarker for things such as immunotherapy response, chronic diseases, response to therapy, and stuff like that. So what makes this problem technically difficult? So bulk deconvolution is difficult because reference single cell expression, so meaning the expression that we know for each of these cell types, is quite variable. And it also comes from a different technology, and it likely has different biological and technical artifacts. The way that we address this problem is by using the concept of domain invariance. In our fruit example, you can imagine that a smoothie was made from an unripe fruit, right? So we have ripe and unripe, and these two domains. And you want to be able to tell me the recipe of the smoothie if it is a ripe or even if it's an unripe fruit. This domain captures spurious correlations that we believe exist in our data set. So ripeness here we believe to be irrelevant to our smoothie recipe. So another example that's kind of like more close to ML tasks is you're given the following training data to predict a camel or a cow. So here we predict camel or we predict cow. This is our training data. It all kind of more or less looks like this. We get extremely high accuracy in our training and our test data. We deploy it, and then now we get a new example. And for some reason, this is predicted as a camel. So why would that happen? Well, it's because of a spurious correlation. So this spurious correlation is this desert background. So actually, what our algorithm was learning was instead the backgrounds of these images instead of just the actual object that we're interested in. So what we're going to do is we want to have a robust prediction regardless of the domain. So no matter where this cow is, even if it's in a place that we've never seen before, we want to be able to detect that it's a cow. So our model learns 3 disentangled latent subspaces to capture the domain, the target labels, and residual noise. So essentially, we believe that the data is generated by having these objects and these backgrounds and some additional noise, and this creates our image. And knowing that having an assumption of how the data is generated, we're able to train a model to help disentangle the targets that we're interested in and remove away all of the spurious correlations, or at least the spurious correlations that we believe exist in our model. So the way that we enforce that these latent subspaces capture the domain, so both the backgrounds and the cell type proportions of interest, we do this through a supervised loss on each subspace. So basically, we have a domain classification error and we have a cell type proportion error. And this entire model is implemented as a variational autoencoder with 2 additional loss functions in addition to the typical VAE loss. So we can kind of see this here. This is kind of in this the cell type proportion error that's here. It's done now in a semi-supervised fashion, so that's why it's kind of in this multicolored version, but I'm not going to go into that in here. So in order to do this, we have to be able to train semi-supervised or supervised a loss on each of these ones. So how do you do that? We need labels. And the way that we do that is by generating pseudobulks. And these pseudobulks, they have known domains and they have known cell type proportions because it's fake data that we're generating. So the cell type proportions, we randomly select the proportions of each cell type and use it to sample reference cell type profiles. So basically, we're just taking single cell references and we're sampling from them. And by sampling from them, from all the different cell types, we know the ground truth proportions. We have like a good sense of the real variability within and between cell types. And then we have our resultant pseudo bulk. For the domain simulation, what we do is we add noise, and we add noise in kind of ways that we expect noise to occur, technical noise to occur within our samples. So we have sample cell type specific fold changes. We vary the number of samples. We vary the sequencing depth. And for the pseudobulk expression, essentially we sum over all the sampled cell type profiles after applying both the cell type and the domain noise. So here's kind of some results on PBMC datasets. Essentially, what we see here are the two latent spaces. So here we have the cell type proportion embedding here, which would be our ZY, and we have the domain embedding here, which is our ZD. And we're coloring them in two different ways. First, we're coloring it by cell type, and second, we're coloring it by the domain ID. So here, each one of these points is a sample, and I color it by the highest proportion of a cell type within that sample. And what we can see generally is that the cell types more or less are able to be closer to one another than to other cell types. And we see that in the cell embedding. So what this means is that our cell type embedding is picking up cell type specific signals, which is what we see as these clusters. In the off diagonal, when we color each of these samples by their domain ID or their sample ID, we see that it's generally diffuse. So meaning that the cell type embedding is picking up the cell type information and not the domain information. And this is what we see again in the domain embedding. So we see in the domain embedding, it's not, each one of these samples has like a high mixture of all of the cell types. We don't see clear clusters within them. However, the sample embedding, each of the samples kind of cluster away from each other. So this is exactly what we wanted. We wanted our cell type embedding to pick up cell types only, and we wanted our domain embedding to pick up domain information only. And here, when we look at just because of space, I only look at cell type proportion estimates for two different cell types. We see that the true proportion here is along the x-axis, and along the y-axis, we have our estimate from BuDDI, and we see very high concordance. So here we have some sort of like descriptive version of our disentangle latent subspaces. We believe that we can take these subspaces and have a strong reconstruction here by choosing different domains. So for example, if we have some sort of perturbation, we'll be able to pick up on these perturbations and learn these transformations from a healthy state to a non-healthy state, or from perturbed to unperturbed. Yeah, and that's it. Thank you for visiting my poster.